Hello, in this video we will discuss about the Calvin cycle. So the Calvin cycle biosynthesis of starch we will discuss. So the Calvin cycle is used for the anabolism, not catabolism but anabolism. So let's begin to understand the major concept about the Calvin cycle. So in the cell which that is chloroplast are present, this chloroplast is the double membrane organelle which can replicate itself due to the circular DNA are present of the chloroplast. So here is the thylakoid membrane which that is used for the inside, used for the light reaction to produce ATP as well as NADPH. So the light will use and oxygen is released from the water. So the photolysis of the water will be occur to produce 2 electron and 2 proton as well as half molecule of oxygen. So the 2 molecule of water will release the 1 molecule of oxygen. So here I am drawing the cell and we are checking where the chloroplast. So first of all I want to say that where the light will be more, so the photosynthesis requirement will be more, if so the chloroplast will move toward the light side in the cell is known as the cyclosis. So here is the cell wall, cytoplasm, chloroplast and vacuole and nucleus. So this movement of the chloroplast toward the light is known as the cyclosis. It is the organelle movement from one place to another within the cell. So during the light more requirement of the light, so the chloroplast will move toward the light side of the sun. So here is the NADPH produce and ATP from the light reaction and the combine with the carbon dioxide to go in the Calvin cycle. It is known as the dark reaction. But the previous is the Z scheme and electron transport chain is the photosystem 1 and 2. Photosystem 2 and 1 which basically used for the light reaction to produce the NADPH and ATP for the preparation of the Calvin cycle by the fixation of carbon dioxide to produce glucose, triose, starch, synthesis and carbohydrate. So yes, C3 plant is very important regard to the Calvin cycle because includes some of the most important source of calories all over the world. 95% of the green plant are C3 plant. In this way, the 3 molecule of carbon dioxide will be fixed in the Calvin cycle we will discuss. For example, rice, wheat, cotton and barley. So let's begin to understand first of all the discovery of the Calvin cycle. So the Melvin and Calvin is the two scientists produce the two students produce the Calvin discover the Calvin cycle within the plant, and it is known as James Basham and Andrew Benson at the University of California to get the Nobel Prize for this work. So let's begin to understand. Here is we will understand from the RUBP, ribulose bisphosphate. Ribulose bisphosphate means the biphosphate. A biphosphate bisphosphate contain a two phosphate group in the carbon number 1 and carbon number 5. So let's draw the picture of the Calvin cycle we will discuss here. So here the carbon dioxide will be fixed in the dark reaction. It is present in the stroma of the chloroplast, not into the lumen of the thylakoid, but stroma. So here is the RUBP. Is the 3 molecule, for example, that is 3 molecule of RUBP, ribulose bisphosphate, will be fixed the 3 molecule of carbon dioxide in the presence of Rubisco enzyme. So the Rubisco enzyme is very important throughout the cycle for fixation of the 3 molecule of carbon via a carbon dioxide. So in this way, after the fixation to dissociate the molecule 6 3-phosphoglyceraldehyde. So 3-phosphoglycerate or glycerate basically, because the negative charge due to the less amount of the proton in the carbon number 1. So this step is known as the carbon fixation. In this way, the 6 molecule you can see and balance the equation. So this is basically the 3-PGA will be formed, which that is the 6 molecule, and 6 molecule mean the 3 into 6 is 18, while the 3 into 5 is also 18. So here is important thing is that the Krebs cycle is also produce the oxaloacetate. For example, the 6 molecule of oxaloacetate in the presence of PCK, the phosphoenol pyruvate kinase. So that is use the ATP, the number of ATP required on the basis of 6 oxaloacetate, so 6 ATP to and release the carbon dioxide to produce P phosphoenol pyruvate and enolase enzyme used to 2-phosphoglycerate and the glycerate phosphoglycerate mutase enzyme used to produce these 3 PGA, and this conversion is also occur due to the mitochondrial Krebs cycle. So the 6 ATP used to 6 ADP produce, and in this way the 6 ATP used to produce another molecule is known as 1,3-bisphosphoglycerate. So this is the reduction, and in this way they reduce the molecule 3-PGA into the 1,3-bisphosphoglycerate. So this 1,3-bisphosphoglycerate will be produced. So anyhow, in this way you can see in this diagram the carbon number 1 and carbon number 3 contain a phosphate group. So after this mechanism, the 6-NADPH is released. So in this way the reduction phase, so in this way the hydrogen will be reduced, the proton will be reduced, and the phosphate group will be released due to the addition of the proton in the carbon number 3. And after the carbon number 3 contain hydrogen ion. So in this way the hydrogen ion cation, which that is the glyceraldehyde 3 phosphate will be formed. In this way this is the 6 molecule again. So this is, you should need to understand about the 6 molecule. It means we are getting the reaction with 6 molecules. So the 6 here, the bisphosphoglycerate is here. 1,3-bisphosphoglycerate will convert into the glyceraldehyde 3 phosphate. It is known as the triose phosphate. This triose phosphate basically is separate the one molecule is known as the glyceraldehyde 3 phosphate. It is used for the synthesis of the starch or other things you should need to understand. So here is important thing is that the ribulose 5 phosphate will be formed. In this way the 6 molecule again, that is basically is the ribulose 5 phosphate will be formed. In this way this molecule is the 3 molecule. So in this way you can see this is the separation and formation of the ribulose 5 phosphate. This ribulose 5 phosphate use the 3 ATP to produce ADP, 3 ADP to form a RUBP again, and this cycle again and again continuously work. So in this way here is the glyceraldehyde 3 phosphate we will discuss, and how to produce the glucose we will discuss. So the fructose 1,6-bisphosphate synthase enzyme used for the 2 glyceraldehyde 3 phosphate to produce the fructose 1,6-bisphosphate. It means the carbon number 1 of the fructose and carbon number 6 of the fructose contain a phosphate group. So that is why it is known as the fructose 1,6-bisphosphate. While the next step is the fructose 6-phosphate. This fructose 6-phosphate is the fructose 1,6-bisphosphatase enzyme used to release of the phosphate group to form a fructose 6-phosphate. So this fructose 6-phosphate is in the presence of it is reversible reaction to produce the fructose 6-phosphate isomerase enzyme. In the presence of fructose 6-phosphate isomerase enzyme to produce the glucose 6-phosphate. So this glucose 6-phosphate means carbon number 6 contain a phosphate group of the glucose. This is the mutation and isomerization. So after the isomerization, the glucose 6-phosphate will be formed, and this basically the NADP plus to reduce into the NADPH, and in this way the glucose 6-phosphatase enzyme used to release of the phosphate group. So after the releasing of the phosphate group, the glucose mature formation, and in this way this glucose is used for the starch synthesis. We will discuss in the next video with the whole detail. While on the other hand, in the case of glyceraldehyde 3-phosphate also can enter again into the Calvin cycle for resynthesis of the RUBP. So how it is possible we will discuss first. Here is the glyceraldehyde 3-phosphate again produce the glucose 6-phosphate. This glucose 6-phosphate is the same reaction are present in the synthesis of starch. So this is the glucose 6-phosphate dehydrogenase enzyme to a dehydrogenation of the NADP. NAD plus will convert into the NADPH, and in this way the gluconate 6-phosphate will be formed. This is the gluconate 6-phosphate decarboxylase enzyme to release the carbon dioxide, and NAD plus convert into the NADPH, and in this way the ribulose 5-phosphate will be formed again. So you can see in this diagram. So I hope you are getting this concept about the Calvin cycle biosynthesis of starch also. So this is the Calvin cycle anabolism.